Hi everyone. I'm Tobias. I'm part of the translational genomics team at Avera Health. Avera Health is a health network in the US based in the Midwest. And I'm going to talk today about how we attempt to offer genome-guided treatment options for our cancer patients, and how we actually also try to utilize open source tools that we have been developing at the Zoo Lab at Scripps. So sequencing allows us to profile the tumor in multiple dimensions. So we can look at the cancer genome to try to find actionable mutations or look for larger rearrangements, for example. We can look at the transcriptome level to find altered pathways or differentially related genes. But obviously all the sequencing data has to be processed first and analyzed in a way that allows for the identification of actually actionable targets that then we can act upon with compound that match to those targets that can be prescribed back to the patient. How does such a process actually look like? So starting with the patient, a tumor sample gets taken. The patient has usually to consent to it. The sample goes to pathology. It gets sequenced, and actually then the data gets analyzed. It gets integrated, and then possible findings are reported back to a tumor board that kind of then decides on a possible treatment strategy for that patient. And this obviously, one aims to try for a quick turnaround for this. We are aiming for 2 weeks. And most of the time actually is taken up here at this part. So this puts obviously a bit of pressure on the computational side of things. So we have to make sure we can turn around the data in a short amount of time. And a lot of those challenges have been addressed here in the last 2 days. We actually want to make sure we automate as much as possible, that we keep track of actually what we are doing and how we are doing things. And obviously we want to integrate those multiple data sources to be able to come up with possible drug recommendations. So the two tools that I want to talk today about are Omics Pipe and OncoRep. Omics Pipe, yay, another pipeline workflow. So pretty much like all the other workflows, able to run sequencing workflows in an automated manner. It's cluster cloud compatible. It runs samples in parallel and has option to keep track of what it's doing. And OncoRep, it's basically a customized workflow within Omics Pipe that adds the ability to integrate data sets, to generate a more comprehensive PDF-based, HTML-based report of the results. It has also the ability to generate more simplified PDF-based reports, and tries to attempts to do some sort of drug matching. Those both are available on BetaBucket. But before I go a bit into the details, let me actually introduce an actual patient sample. So this is a 69-year-old female with metastatic ER-positive, PR-positive breast cancer. She's been diagnosed initially 20 years ago. She's been progressed into a 4 and to a 6, and she, as you can see, through the course of her disease, she went through an impressive list of different treatments. And the last one being tamoxifen in 2012, end of 2012, and a PET scan in 2013 showed recurrence of disease. So she had multiple metastases in liver, the lymph nodes, and the bone. I'm going to come back to her at the end of my talk. I think what I kind of a bit want to highlight about Omics Pipe is that we implemented published best practice pipelines into it. that can be pretty much run out of the box. So there are two RNA-seq-based pipelines, one of them the tuxedo suit. There's GATK-based variant calling. This is actually OncoRep. And then we got two SHIP-seq pipelines and two microRNA-seq-based pipelines. One can run a workflow based on a YAML config file that basically has a list of samples and also config options for the various tools within the workflow. And then obviously Omics pipes will handle the distribution of the jobs and the parallelization. We use Sumatra to keep track of the run, which kind of stores in a database things like run ID, executables, parameters, and so on and so forth. As I mentioned, OncoRep is a customized workflow with an Omics pipe. Right now, it's RNA-seq based only. We're working on extending it to the DNA level. It does RNA-seq based variant calling, fusion detection, and gene expression quantification. Obviously, QC metrics will be generated. Variants that are called will get annotated. We try to prioritize them in a way that we can select for possible driver mutations, or the called fusions. We use a tool called OncoFuse that kind of tries to predict the oncogenic potential of a detected fusion. And on the expression level, for breast cancer, we generated a reference cohort based on TCGA data and normal healthy controls. And the new patient sample is kind of scaled towards that reference cohort using a add-on normalization procedure. And that actually allows us to generate predictors, for example, for molecular subtypes of breast cancer or for the receptor studies on that reference cohort and transfer them onto the patient. We use the normal healthy controls to do differential expression analysis, and those differentially expressed genes go into pathoanalysis. And in the end, we'll hopefully gather altered pathways, altered genes, the predicted molecular subtypes or receptor statuses for breast cancer. And those results get matched against TRAC databases. Right now, our resources, our main resources, we rely on a TRAC bank, CakeTRAC, and PharmGKB. And all those results are, in the end, kind of accumulated into a HTML-based report. This is basically generated using knitr. All the results are kind of browsable. Tables are exportable into spreadsheets, for example. We try to link back to genes to like GeneWiki or NCBI Gene, and so on and so forth. But we have also the ability to generate a more simplified report that, for example, could be handed to a treating physician who, as Malachi earlier pointed out, they usually don't have a lot of time to go into depth into those analyses, so they're usually more interested in, hey, what's the target? What are possible drugs that I could target that with? And what's the evidence? For example, is this target structure a gene that might be upregulated, or might carry a mutation, or is part of an altered pathway? Those reports are also fully customizable. We use SWeave to generate them in R. As I pointed out, I wanted to come back to that actually patient. So we run her data through those two tools, and we were able to accurately predict her receptor status, so it's aligned with pathology. We found 35 up and 27 downregulated breast cancer relevant genes. Among them, well-known proliferation and progression markers like Ki67, cyclin D1, aurora kinase A. We saw upregulation of genes that might explain the resistance to tamoxifen treatment. So remind you that was the last treatment she actually had received, and another marker that is associated with poor outcome of that patient. But among the differentially expressed genes, there were also new candidates that might be might be useful for new treatment option for that patient. So there was an option for a platinum-based chemotherapy or additional targets where compounds could be added to a chemotherapy, for example. So this was basically what was suggested to the tumor board. There was the list of compound that was suggested, which obviously, and I think Malachi also kind of mentioned that a bit too, leads into another problem that although you might predict a list of possible compounds, not every of those compounds might be accessible because often they are still under investigation. They might be you might be part of clinical trial to have access to a compound, or the insurance just might not pay for the treatment. Maybe unfortunately, I can't present anything on the outcome for that patient. I couldn't follow up on her, so I'm sorry about that. But at the end, obviously, I really want to point out Katie. She's been spearheading the development behind Omics Pipe, and thank you for your attention.